by the Society for Experimental Biology and Medicine, SEBM, and its Society Journal, Experimental Biology and Medicine, EBM. The SEBM is one of the oldest scientific societies in the United States. It was founded along with its journal in 1903 and has had many distinguished members over the years. Hi, Josef. The society is multidisciplinary, as best evidenced by the journal's 21 research categories ranging from anatomy to synthetic biology. Membership in the society has many benefits, including no page charges when publishing in the society's journal, EBM. Under Steve Goodman's leadership, and Steve is the editor-in-chief, the journal has an impact now of greater than 3, and even though submissions have increased substantially, it accepts only about 25% of submitted articles. One of the SEBM's priorities is the encouragement and involvement of young investigators in the society's activities. Thus, the question and answer session of each of the webinars is moderated by a postdoc or advanced graduate student. Today it will be Tori Golub from Texas A&M. We have 2 speakers today, Dr. Josef Penninger from Vienna and more recently from Vancouver, British Columbia, the University of British Columbia, and Dr. Naama Geva-Zatorsky from the Technion in Israel. Our first speaker will be Dr. Penninger. He's a highly regarded virologist and cell and molecular biologist geneticist who recently was recruited to the University of British Columbia in Vancouver. And he was recruited from the IMBA, the Institute for Molecular Biotechnology of the Austrian Academy of Sciences in Vienna, where he was the scientific and founding director. He was recruited to UBC as the Canada 150 chair in functional genetics and as the director of the Life Sciences Institute. His research areas are many and are epitomized by a phrase on his lab website that I particularly like and can relate to. And the quote is, "We are not afraid to wander off the beaten track and merge ideas from apparently disparate fields." I can say that's also true for our other speaker, Dr. Geva-Zatorsky. So in today's talk, Dr. Penninger will speak about ACE2, which is a peptidase in the renin-angiotensin system. It is a partner in amino acid transport, and most relevant for today's talk, it is the receptor for the SARS-CoV-2 virus. He will, I believe, speak to the potential of using soluble ACE2 as a decoy to reduce viral load. What's interesting is that Dr. Penninger has also observed and published that ACE2 functions in the intestinal tract and plays a significant role in maintaining the ecology of the gut microbiome. This provides a nice segue for the introduction of our second speaker, Dr. Naama Geva-Zatorsky, whose main research focus is on the gut microbiome and its interactions with the host immune system as it relates to overall health and disease. As you will hear, she and her laboratory have also developed a very rapid point of contact isothermal assay for the detection of SARS-CoV-2 RNA. Dr. Geva-Zatorsky spent 6 years as a postdoc at Harvard Medical School and then moved back to Israel and set up her lab at the Technion, Israel Institute of Technology. She recently co-founded and is chief scientific officer of BioTax, a new startup company. and I presume it will focus on gut microbiome and health. She's the recipient of numerous honors and awards, including the Horev and Alon Fellowships, UNESCO's L'Oreal Award, the John F. Kennedy Prize, the Teva Prize, and the Baron-Holtz Prize for Academic Excellence. I think that's enough for both of the introductions, and I'd really like to get started with Dr. Penninger, please.
Good, so it's up to me. Share my screen. Okay, so nice to meet you online, and thanks so much for the introduction. So I get right into my story. So it's about how we discovered ACE2, to describe to you what ACE2 is doing and how this relates to the first SARS outbreak and COVID-19, and how we learned to develop a medicine based on this. So that's my new playground. Already mentioned, I'm running now the largest life sciences institute in Canada at the University of British Columbia. I still maintain a little lab in my old place in Vienna, just to finish up of some projects, but I have still an active research program. So 22 years ago in my lab, when I was a young investigator in Toronto, we were looking into heart development of Drosophila. So we were really curious if we can find genes which regulate heart development. And eventually we found some. So this is just of some markers called Eve and Tinman. And these are the heart tube formation, and you see the different cells. But what we actually found was one of the hits was very close to ACE, angiotensin converting enzyme. And then we realized flies had 2 copies of it. But in humans, of course, we knew ACE for a long time, but there was only ACE known. So we thought if flies have 2 copies, then there might be a second ACE. So Mike Krakau in my lab cloned the second ACE. That's why we call it ACE2. It's a transmembrane protease. Here was the first expression in the mouse. It's in the kidney, in the lungs, in the heart, in other tissues. And it's very similar in the catalytic domain to the classic ACE. I should also acknowledge we were not the first to publish ACE2, the sequence. There were 2 other groups. We had it in our fridges. The reason why we wanted to not published yet at this time was because we wanted in vivo evidence. So we made mutants and mapped the pathway. So just to remind you on the renin-angiotensin pathway, what had been known, angiotensinogen is cleaved by renin into angiotensin 1, a decapeptide, and ACE, the classic enzyme in the pathway, cleaves the last 2 amino acids to make an octapeptide called angiotensin 2. And this regulates vasoconstriction, sodium reabsorption, cardiac hypertrophy, heart failure, hypertension, many, many diseases. But there's a huge set of literature out there. H2 drives inflammation in many diseases. So this is where the inhibitors, of course, come in, the ACE blockers. ACE knockout mice have hypotension, they have kidney pathology. And this is then where ACE2 came in. ACE2 initially was identified to cleave 1 amino acid. So from Ang1 to angiotensin 1 to make this nonapeptide called angiotensin 1 to 9 with no real function we know yet. And also can clip Ang2, the last amino acid, to make angiotensin 1 to 7. So the second thing we did, we actually mapped it to hypertensive rat strains. There's 3 of them, spontaneous salt-induced hypertension, and ACE2 mapped right into an EQTL. And this is also important for COVID-19 because it's on the X chromosome. There's a massive gender bias in severity of disease, and maybe ACE2 being on the X chromosome could explain this. So this was the first phenotype we ever found when we made the knockout mouse. So we made the first knockout mouse published in Nature 2002. And this is what we saw. If you knock out ACE2, they get impaired, spontaneously impaired heart function in this particular mouse background. If you challenge the hearts for hypertrophy called TICC, transaortic bending, constriction, ACE2 knockout mice get very severe disease. So here's our genetic slang, ACE2 minus Y, because this is a... And when we knock out ACE on an ACE2 knockout background, and we see total rescue of the phenotype. So basically what we discovered in vivo, ACE2 is the negative regulator of the system. It balances out the levels of angiotensin 2. So ACE makes Ang2, ACE2 gets rid of Ang2. With all the downstream consequences of Ang2 driving disease, heart function, kidney disease. ACE2 is also a very unspecific protease. It cleaves other peptides at very high catalytic activity like apelin, bradykinin, neurotensins. So, and then all of a sudden we found ACE2 in the lung and started wondering what ACE2 is actually doing there. So in normal lung, if you knock out ACE2, nothing really changes. So we wondered under these conditions of lung disease. So this is really work of Yumiko Imai. We got into this already 2002 and built an intensive care unit for mice. There were maybe 4 or 5 groups in the world who actually were doing this because very complicated, a stable system to study lung injury and lung failure in mouse models and have reproducible data. So Yumiko actually pulled it off. So this is the model. We had an acid aspiration model for acute lung injury, which changes elastance, which is a measure for lung function. There's severe lung edema, there's MPO activity, so inflammation here, the histology. So there's massive bleeding, the blood vessels open up, edema formation, albumin spills into the lung. So a model for acute lung injury. Then when we looked in ACE2 knockout mice, what we found is the mice get it as super severe disease. So if we inject actually blue color into the belly of this mice, it's so bad that all the blood vessels start to leak and all the blue color comes out in the lung. So the knockout of ACE2 leads to very severe lung injury, much more severe than a wild type animal under conditions where you actually trigger the disease. So we also mapped it. So it turns out this is a catalytic function and function of Ang2, because if we knock out ACE on this background, we see rescue of the phenotype. So this was very nice because we had worked out basically a completely new function of the renin-angiotensin system in lung and unspecific triggers of lung injury turn on Ang2. If you take away ACE2, then it gets much worse because the function is to balance it out. And then SARS hit the world, 2003. This is SARS over Toronto, and around 8,000 people, to put it in context to COVID-19, 8,000 people were infected with SARS-1, around 400 died. It took around 1 year from the first outbreak to ending it, and SARS-1 was not very infectious. So at this time I was working in Toronto, just moved to Vienna, hoped I had escaped the virus, and then it hit me because Mike Fasan from Harvard actually published that ACE2 might be the receptor for the spike glycoprotein of SARS coronavirus. The spike would bind to ACE2, and by doing so gets entry into the cells. However, there were many other potential receptors identified, like L-sign, others. So it was not totally clear if ACE2, of course it binds, but it would be an essential receptor for the SARS coronavirus. Since we had the first knockout mouse, and actually I think at this time the only ACE2 knockout mouse, we sent our mice to China. They got infected with the mouse-adapted strain, and then the experiment came back. In wild-type mice, the virus replicates. There's a little lung disease here, a little injury, not like in humans, but a little disease. And if we knock out ACE2, the virus cannot grow. So this was the definitive proof that ACE2 is the in vivo receptor for the SARS coronavirus. No ACE2, no infection. The other thing we saw, if we infect mice with the virus, is this Western blot in lung and heart for ACE2. And if you see after infection, the protein disappears. So the virus actually leads to an animal which is like a knockout mouse. And so could this actually explain why the SARS and now COVID-19 viruses became killer viruses? Because they hit a molecule which normally protects from lung injury. and injury of other tissues. So we set up a model system where we cloned the spike from the SARS coronavirus, made it in our model with acid injury on the bottom is normal lung, acid injury leads to disease already. And just by putting in the spike in the system, it made it much, much worse. We cloned the minimal binding site, so only this region of spike is sufficient to make lung disease worse. And this is, of course, the region which binds, it's the receptor binding domain of spike binding to ACE2 is found now in multiple cryo-EM and crystal structure studies. So we came up with this model, which it seems now every company and every review is using on the issue of COVID-19 and ACE2. So in unspecific triggers of lung injury, pneumonias, different triggers, acid aspiration, sepsis, the renin-angiotensin system gets turned on, and via the AT1 receptor drives severe lung injury, lung edema, probably also via bradykinin receptors, inflammation, you name it. And ACE2 balances this out. If you don't have ACE2, the injury gets much worse. Now the virus comes, binds to ACE2. Together with ACE2, it actually gets into the cell. ACE2 gets degraded and downregulated. ACE2 is lost from the surface, and therefore the disease happens. So a molecular explanation why SARS became a killer virus, because ACE2 plays a protective role in acute respiratory distress syndrome, and SARS corona spike protein downregulates ACE2, and by doing so enhances the severity of the disease. So an explanation why the SARS virus hitting a particular ACE2 drives this very severe pneumonia. So could we use this as a therapy? We cloned human recombinant ACE2, put it in our system in mice. This acid injury, the red arrow shows when we add our molecule, and you can see it stabilizes disease. So based on all of this, so ACE2 being the SARS receptor and protect from lung injury, we developed, starting in 2005, 15 years ago, human recombinant angiotensin converting enzyme. A soluble form of it is a therapeutic use, basically as an enzyme replacement therapy to do this. So it's conflict of interest. I have to declare that's a company I started at the time. So the ACE2 molecule is, we did all stability. This went actually through 89 humans in phase 1 and phase 2 clinical studies in lung disease. It's around 10, 12 hours left, depending on species, human 12 hours. It's stable. We can freeze it, we can sow it, we can send it all over the planet. There's of course a lot of issues in drug development one has to consider. Stability, production, that everybody gets the same dose of the protein. So this was a piglet experiment, acute respiratory distress syndrome in response to LPS. We give a bolus injection of our drug, which we call APN-01, and we can stabilize oxygenation. So the real idea why we developed this was, of course, there are many reasons which lead to the syndrome we call ARDS, acute respiratory distress syndrome, which of course the end syndrome of many diseases like sepsis, aspiration, gastric aspiration, SARS coronavirus, the Spanish flu, for instance, bird flu, bioterrorism, you name it, smallpox. Just because I used to live in Vienna, of course, I have to show a painting of somebody who died of the Spanish flu, Egon Schiele. As you know, there was probably 30 to 50 million people who actually died. And the Spanish flu actually took, I think, 3.5 years. It went in waves around the globe until it stopped. So I think with COVID-19, we have to anticipate something similar. That's another paper we published together with colleagues in China. So that ACE2 protects, for instance, from lethal avian flu infection. So clearly this principle that various triggers of lung disease can be alleviated with ACE2 holds true. So this was fine. I actually used to be Mr. ACE2 in the 10 years ago. We wrote all the reviews. We published tons of papers. It's all in the textbooks. But of course, then the interest waned because there was no SARS disappeared, and so what was the big deal then with ACE2? Beside, of course, its basic physiology. And then, of course, SARS coronavirus 2 hit, triggering COVID-19 and this pandemic. Based on the first sequence, which was published very fast, it was absolutely clear ACE2 must be the receptor because it had high homology at the spike protein to the first SARS coronavirus. And briefly after, in multiple papers, it was clearly shown that ACE2 is the critical SARS coronavirus receptor, as predicted from the sequence and what we had shown 15 years earlier for the SARS virus 1. So ACE2 is the receptor also for the new virus causing COVID-19. The structures were solved very fast, so we know exactly how it looks. On the top in green is ACE2, this is the spike protein. Here I just show it, we've never published this. ACE2 as well as the spike protein are heavily glycosylated. So people considering vaccine development need to consider glycosylation. And to be honest, I'm quite worried when people actually do vaccines and shoot RNA into the muscle, because the glycosylation of the spike in muscle might be very different what you get from a real virus. So let's see how this actually works out in real people. So we should be very careful with the vaccines we develop because of different glycosylations. And of course, we don't really understand yet how the immune system acts. So what we recently did, we asked 2 questions. Can soluble ACE2, the molecule APN01 we had developed over many years for lung disease, can reduce SARS coronavirus infection? So the reason is this, because these 2 viruses, the first and the second SARS virus, bind, of course, to ACE2. This is the signal for internalization. There needs to be a clip via a very unspecific protease. 2 proteases, one is called furin and another one is called TMPS2. But they're unspecific, so the real receptor is ACE2, and when it binds, then there's a clip by the protease, so the spike goes down to the membrane and enters the cells. So this is via endocytosis, it goes into the cells, the virus, of course, spills out, which triggers the diseases SARS and COVID-19. So this is the molecule we developed, the soluble form of it, and it has very high affinities, like a high affinity antibody. The affinity is 1.2, 1.6 nanomolars for the spike protein. So the idea is, of course, soluble ACE2 would function as a decoy, soak up the virus, and so the virus cannot find its real receptors and have improved disease. So like a very specific antibody, it would block the virus to enter the cells. And of course, you know, if you have a monoclonal antibody, all you need is a mutation in the virus and the virus will wriggle out from the detection. The virus cannot escape from ACE2 because it makes a mutation to escape from ACE2, then it cannot infect our cells anymore. So that's why this might be a reasonable approach. We hooked up with Ali Mirasimi in Stockholm. He isolated from the first Swedish patient, you see on the 7th of February, the virus. So this is how actually the virus we are using. It's this first Swedish isolate the virus. And this is the experiment. Ali's team in Stockholm, it's this high security Northern European viral laboratory. They work on Ebola and other things. This is where we do our research on the virus, or the infectious features on the virus. So we took VROE6 cells, infected them with different numbers of the virus, here by Moy, 10 to the 6 to 10 to the 3. And then we gave them recombinant ACE2, human recombinant ACE2, and as you can see in the dose-dependent manner, it reduces the viral load by a factor of 1,000 to 5,000 times. If you use mouse recombinant ACE2, it doesn't do anything because this mouse ACE2 actually doesn't bind to the new virus. So indeed, soluble ACE2 can function at least as good as remdesivir, probably even better to block the virus load. We're just writing a paper to compare the 2 drugs. The other issue is, of course, ACE2 is expressed on multiple tissues. We know COVID-19 starts in the nose and the throat, goes deep in the lung. This is where ACE2 sits. That's why people get this typical pneumonia. If this pneumonia, of course, gets severe, it spills out into the tissues. And we know late-stage COVID-19 is not a lung disease. It's a multi-organ failure disease affecting many tissues. And of course, ACE2 could explain it because it's expressed all over the place. It's in the heart, it's in the blood vessels, it's in the central nervous system, it's in the intestine, as we had shown in a paper in 2012. It's in the kidney, in the proximal tubules. ACE2 expression, and there are lots of studies now going in, changes with age, with gender, with cardiovascular disease, with diabetes, with smoking. So I think this we have to figure out. And ACE2 could be certainly also variants in ACE2 could be certainly an equation by some people get it worse and some people get less worse, by certain tissues are infected, by people with cardiovascular disease, diabetes, smoking are differently affected. So the second question, of course, is can soluble ACE2 protect from organ damage? As I had shown you from the lung injury, which is a catalytic function of its enzyme function in the renin-angiotensin system. So just digging out some old papers we published, this is a heart disease model, and we give recombinant ACE2, so that's actually the same molecule in humans. In the blue, dark blue bar, it always gets better. So published in many papers by many groups. A liver fibrosis, recombinant ACE2 makes it better. Kidney disease, actually turns out older male mice get spontaneous kidney injury, older females get it much less, which I find really interesting in the gender bias we even see in mice which lack ACE2. Here's kidney diabetic mice with kidney disease. And again, the blue bar, if we actually substitute and give mice this APN01 recombinant human soluble ACE2, then it gets better. So it's always the same, based on the basic biology. If we take away ACE2, these diseases get worse because Ang2 drives disease. Fibrotic disease, kidney disease, heart disease, hypertension, also thromboembolism, so, which already was known. And of course, ACE2 is the negative regulator of this. You take it away, it gets worse. You have this disease model, so supplement ACE2, it gets better. So it makes all complete sense. So we actually, to study this directly, we developed human kidney organoids and human organoids, which we just published recently, to really show if human tissues, or we can use engineered human tissues for infection. Let's work with Nuria Montserrat in Barcelona. She makes these beautiful kidney organoids from stem cells, and the proximal tubules where ACE2 is expressed here in green. And here's single cell analysis of ACE2 in proximal tubule cells, also in podocytes. So if we infect this engineered human kidneys with the SARS coronavirus 2, so it replicates in the kidney, we can find this viral RNA, we can actually find infectious virus, viral progeny, and all of this can be blocked with recombinant ACE2. It's the gate to enter the cell. Obviously, if we add it, then we block the virus to get it. We did the same in gut organoids. Actually, we had it in our first paper, but we were told by the referees to take it out. But there are now 2 papers published in Science and also Nature Medicine to show this. The gut organoids, they've heavily expressed ACE2, which we published 2012 in Nature. It has a different functions, actually controls a single amino acid uptake, stabilizing a transporter called BOAT1 in a disease called Hartnupp's disease. There are mutations in it, and clear enough, the virus can replicate in gut organoids. There's ACE2, obviously can get in. And people with COVID-19 have diarrhea, and one can find the virus in this. The other thing we did, we engineered human blood vessels, which we published last year. So the perfect human vasculature, capillaries with lumen, pericytes, endothelium. I'll make the story short. We can infect this with the virus. We have ACE2 actually on pericytes and endothelial cells. We can recover virus. We can recover infectious virus. And this again can be blocked with ACE2. And we have now similar data actually with lung organoids. Same story, we can infect lung organoids. They have ACE2 and we can block with recombinant ACE2. So this is just my second last slide. So just to remind you again, COVID-19 is a multi-organ disease. And what has emerged recently is really that some patients, maybe many patients at later stage have very severe infection of the blood vessels. This explains why some patients get strokes, probably why some kids actually get Kawasaki-like syndrome. And of course, our data explain it. So we have ACE2, the virus can directly infect the blood vessels. So this is also important because if the virus spreads, how does it get into other tissues? And we know it's in the kidney, it's in the gut, it's in the liver, it's in many tissues. And of course, it has to go through the blood vessels. So now we know it can directly infect them. You can get infectious virus, and of course it can, by doing so, get into the tissues. So multiple tissues in the disease are affected. So it's a very unique disease. ACE2 is actually in the brain. Some people cannot smell, taste anymore. What I find really interesting, some patients are blind to hypoxia, so the virus switches off the hypoxia sensing, so it generates basically people who are very sick, spill the virus, but believe they're healthy. Of course, it's a perfect way for a virus to spread. We know people with cardiovascular disease, kidney disease, diabetes, the ones which will get very severe disease. So again, all these diseases deregulate ACE2 expression. So ACE2 can explain a lot, of course, not everything, the immune system, the aged immune systems, the innate immunity, but it explains clearly the way of infection, explains the spreading of infection. It can also probably explain severity of infection in certain conditions. And based on all of this, we're actually now in a phase 2 placebo-controlled double-blinded clinical study in Europe, actually not in China. So we're talking still to the Chinese authorities. The trial has already started in 4 countries, and we're actually trying to get more countries now on board. And the reason why we do this, so APN01, recombinant soluble ACE2, and we got the approval from the EMA, and we're actually talking now to the FDA to move this also to North America, is it has 2 functions. One is it blocks the virus like a very high affinity antibody. So it reduces the virus infection. And at the same time, engineered in the same molecule, it protects the organs from organ damage via hyperactivated RAS and angiotensin II. And these 2 functions are independent of each other, engineered in the same molecule. One side binds to the virus, the other side has the catalytic effect. So that's why we're testing it. And I'm actually just writing up the first report on compassionate use in patients, which looks really promising. And with this, thanks for listening to me. Mike Krakauer cloned ACE2, made the first knockout mouse, phase 2 in the world, Yumiko Imai, Keiji Kuba, together with intensive care physician Geva-Zatorsky in Toronto, really made the first link to lung disease and linked SARS to lung injury. In many people helped us, Nuria in Barcelona with kidney organoids, Rainer and Alex with blood vessel organoids, and all our infectious work we do in Stockholm. The person who does the real infection is Vanessa in the high security Northern European laboratory of Ali Mirasimi. Thanks for listening.
Well, thank you very much. That was fantastic. I use the word fantastic because I learned a lot, and I really appreciate the talk. We'll now move on to Naama, Geva-Zatorsky, and she will tell us a little bit about what LAMP is and when to take over from here. You're muted.
Yeah, exactly. Looked for the mute button. So yeah, thank you so much for inviting and giving me this opportunity, and happy to see you all, and it was really fascinating to hear Joseph. I will downplay it because this is, as opposed to Joseph, for me, this is really off track of my day-to-day. I will explain a bit more later, but as Peter mentioned, and thank you for some kind introduction, I usually study the gut microbes and the host immune system. Now, perhaps Joseph and I I'd be happy to talk about the ACE2 in the gut. And this project was off track. We were sitting at home and brainstorming the whole lab for days and thinking how can we help to this pandemic. I will continue the sentence in a couple slides, but that we came up with the thought that we should work on a rapid detection of the virus. So the need is spelled here. Many patients are on the doors of the hospitals trying to asking for detection of the virus. It's more than the capacity of the hospitals around the world. Most of the patients in Israel were negative at Rambam, the hospital next door to us in the north. The rates were at 2%, even when the pandemic was at the peak. Maximum 4% of the patients that came to the ICU, so they were very sick. So it's a large burden on the hospitals to do all these tests. A lot of professional people, professional team, and very expensive. It takes time, etc. I guess I don't need to explain much because we all experienced it. So that led the Israel and other countries to give a list of criteria to who is allowed to be tested, and still it's quite a lot of burden. So I'll skip the slide. And the idea was to look for try to develop a test that can be easy and cheap, and can be tested at home with less professional team to reduce this burden on the healthcare. So so now back to the sentence I started. Actually, these two friends that are actually currently in Boston, Shai and Asaf, I called them with an idea for them for the COVID-19, and they replied with, put that aside, how about this? And it took me a couple of days to think about it, and then I called Professor Michal Powell, which is the head of infectious disease at the Rambam Hospital, and she immediately said, yes, 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 we have to do this as fast as possible. And all this, the team in the Rambam was amazing. Moran, the head of the virology lab, Yuda, the head of the research, and the head of the GI, and Ronit and Shimon. And really, this was part of our power, this immediate call. Everyone stood up and helped us to get Helsinki approvals and access to samples. And the team in the lab was also amazing. It was quite an experience. It's actually, what I will present to you today was a work of, an actual work of about 2 weeks, but 2 weeks of 24/7. We all came to lab, and as you can see, we were, at least I can tell that I look tired here. We really slept on the sofa in lab for like an hour and a half, and not every night. So it was really, really crazy because we felt we can, the motivation was to try to help as fast as possible because we saw this works. So in a normal PCR reaction, I guess I'm sure I don't need to explain. It's an exponential growth of the product, but it requires a sophisticated machine, lab, and professional team. In lab, there is a really cool trick that I think the movie will show it. So I would like to show you the movie from NEB Labs that I will actually elaborate in a moment. So yeah, it will work in a bit.
Is it loading?
I just realized you didn't see the movie, right? Okay, let me. I will show you. I think the movie is important. If it's a, what do you think, Peter?
If it doesn't take too long.
Okay, one moment. Sorry. Sorry, I'm sorry about this. Just one moment. I can also just give you the link if you, whatever you prefer. If I do this, I think it will work.
There you go.
Okay, sorry. I did the share. Yeah, okay. Thank you. Sorry about that. This is from NEB, the NEB Lab website, and actually the reaction mix that we use here is from NEB. And I didn't emphasize it, but Rich Roberts and Nathan Tanner from NEB were very supportive with any questions, with all the questions we had along the way. It was really a nice and fun endeavor together. So the importance, the LAMP allows us to perform a PCR reaction and amplification of nucleic acids in an isothermal reaction without a PCR machine. You don't have to do these cycles of different temperatures at different time intervals, and it grows even faster than PCR. The RT LAMP mix that from NEB is also colorimetric. So in the PCR reaction, like in other sequencing reactions, protons are released, and this indicator, the pink indicator, turns yellow when the pH is reduced, when protons are released. So NEB actually already published before we started, and that triggered this project. NEB published Zhang et al. I think I have the citation in a couple of slides, and Nathan Tanner, that with this reaction mix, they can detect a nucleic acid of the coronavirus, of SARS-CoV-2, but they did it with synthetic nucleic acid and purified RNA. So we were interested to test whether this can be applied to actual samples directly from the sample material. So we first tested positive and negative samples. We inactivated the virus. We inserted the lyzed virus to the LAMP reaction mix. The reaction is at 65 degrees for 40 minutes, and the results are colorimetric. So these are negative tubes, and these are positive. The first experiment was on purified RNA. We received from the virology labs at Rambam purified RNA from patients that are positive and patients that are negative, and this is the non-template control. Primers, we used the primers from Zhang et al. From the publication of NEB, and it was very clear that the positive sample was yellow, the negative sample was pink. We then tested samples. So the first and the majority of this project until now was on swabs, because these are the samples that we had available and the Helsinki approval for quite immediately. From swabs that were submerged in the universal transfer media, we took a couple microliters and immediately performed the reaction. I mean, we boiled for 95 degrees for 5 minutes, and then added a couple of microliters to the LAMP reaction. And even directly from samples, we can see positive samples were positive and negative samples were negative. The first protocol was the direct protocol. It was not good enough. Here we tested 100 swabs. Actually 70, so I'll walk you through this graph first. The x-axis here is according to the qPCR results of the hospital. So the standard qRT-PCR test, and the numbers here relate to the CT, the cycle where the RNA comes up. And I'm sure all of you know, just to recap, lower numbers or lower cycles meaning higher viral load, and higher numbers mean low viral load. So 70 samples were negative, and none of the negative samples came up negative in our reaction. So positive and negative in the RT LAMP are shown here. Only too few positives came up. So we weren't happy enough. We tweaked just a little bit with adding proteinase K and guanidine to the reaction, and some improvement of our results. I'll show you all the results in a moment. And we also were interested to test the specificity. So we compared swabs and both purified RNA and samples directly from patients that were infected with other viruses. So always T = 0 is the pink, what's the color of the sample at the beginning, and T = 30 is after the RT LAMP reaction. So the first here are HSV swabs, HSV purified DNA, RSV purified RNA, influenza B, RNA, enterovirus RNA is 6. RNA extracted from SARS-CoV-2 positive patient is the only yellow sample, and no template control. So I would like to point out that some of the samples are not dark pink at T = 0, and that's why this is, well, anyway, this is not a yellow like number 7 here, but I would like to have you pay attention to the fact that some of the samples are not dark pink at T = 0. And this is even a bigger concern when talking about saliva, but we will talk about it in a moment. So yeah, and I'll talk also about image analysis in a moment. So the only positive sample here was the SARS-CoV-2 RNA. The rest are considered negative, they're not yellow. So then with the improved protocol, we tested almost 90 patients, swabs again. This is the same graph that you're already familiar with. So here we got positive results for over 96% of samples that had a medium to high viral load, so cycle 28.8 and below. These are the positive ones in LAMP, and this is the 28.8 number. Some were not detected positive. We show here one false positive, but it's actually just because we were very cautious. By eye, it wasn't yellow, but we worked in the virology BSL2 plus room at the hospital. All the results, we took pictures with our phones because any machine that we bring there would be contaminated. So we were very cautious. So by eye, it wasn't yellow, but afterwards in the pictures, when we passed it around the lab members, some of them considered it positive. So this is the false positive that we report on. Regarding the timing, we compared 30 minutes of the reaction. So the reaction itself is 65 degrees for 30 minutes. So this false positive one came up at the very beginning and didn't change throughout time. So it's not that we saw more false positives at 40 minutes. And at 40 minutes, the percentage was the highest. So if we look at cycles that have low viral load, we don't see true positives. At medium viral loads, at 40 minutes, it's an 88% success, and at high viral loads with cycles that are lower than 26, we have at least a 96% success at 40 minutes. And these are the numbers. So at 40 minutes, let's focus on this one. We have one false positive that came up already at 30 minutes, and the true negatives were 96%. True positives were 80%. It depends on the cycle, as I mentioned, and false negatives were 19%. Depends on the cycle. If we look at only medium to high cycle, then it's higher. But the holy grail for us was directly from saliva, because if we are interested in a home test, swabs will be difficult. So we got approval for a few saliva samples. So it was really like they approved us to get a saliva sample, preferably from a doctor that was already confirmed to be sick. So we searched around Israel, found one, and tested him and his family. So let's start at the bottom. So these are the patients that were confirmed to be positive, and in our reaction, they were yellow, positive. This is a negative control. So the negative control was negative, not yellow, and we performed the standard qPCR from the saliva, and these are the numbers that the cycle of these samples. So a very high viral load from the saliva. Actually, after a couple of days later, there was a publication on a study on saliva versus swabs that showed that saliva is more sensitive than swabs. And here, I can't say for sure, but we all felt that these high numbers mean that the saliva is probably more sensitive than swabs. But this is the lamp, which is yes or no at this point. Yellow is yes, pink is no. No primary control. And POP7. POP7 is primers for a ribosomal gene in humans, just as a positive control for the reaction. So the idea is that even at home, there will be reaction mixed with the positive control, so that the person will know that the reaction worked. So if the positive control become yellow, hopefully also negative control that remains pink, and then the test itself that will either be similar to the positive control or hopefully similar to the negative control, so that anyone can compare the pink, yellow, and decide. So so this is the reaction, and we actually tested it in a thermal cup in the lab. So the person spits into a tube, then the tube that already contains proteinase K remains in the room temperature for 15 minutes. Then a deactivation step, which we really performed in a thermal cup just to make sure it can be done. So just hot water from the bar at the end of the corridor, put the tube in, 5 minutes. Then we added cold water to reduce the temperature to about 65 degrees. It doesn't have to be precise. And the reaction for 30 minutes, 30 to 40 minutes in 65 degrees. Negative is pink, positive is yellow. I'm laughing because the first talk with Rich Roberts from NEB, we asked what does he think about protein SK, and said yes, and we were like, how should we start? How many microliters? What concentration? And he's like, I think just a little bit would be enough. And it really, like a grandma recipe, just a little bit protein SK, plus minus 65 degrees, and that's why I think it's very applicable for homes. But still, it's better if we will develop this on a device. So the dream is to have a device where the person can lick or just preferably lick, but maybe just a small spit, and that's it. And then do nothing, and the device itself will turn on the temperature to take all these reactions very simply, and then after 40 minutes, the person can look if the reaction became yellow or pink. So I would like to say also that we did some image analysis as well. So we're testing whether there are shades of pink. Maybe I'll go back to these. As you can see here, well, this is negative, but and here it's not. Well, anyway, I won't move too many slides, but here it's very clear that the yellow is yellow and the pink is pink. But we did some image analysis so that the app that made perhaps an app can actually read the results, and then the person does not need to make the decision himself and it's herself, and it's worked very well. And now we're testing whether there are also shades of pink, and that maybe will allow us to detect the lower viral loads, because we only consider yellow as positive. So another thing we're doing now is to test whether we can quantify the pH in a different method, and that perhaps will let us detect lower viral loads, because as soon as the protons are released, the pH starts to change, and but the motto is that the idea is to have a very simple test. So we don't want to have a very expensive and sophisticated machine, but as simple as possible. Patient can only either put this loop in the mouth or spit somewhere or lick something and see the difference in the color. Peter asked me to talk about about other tests that are out there. So many of the tests, there are many tests, and actually every several hours I see a new publication. It's really amazing how the world did everything people can to help in this pandemic. So some of the home tests are based on antibodies. Antibodies come up several days after the viral infects us, can come up several days after the viral infects us, so we might not detect the early stages. And also antibodies can stay after the viral is already cleared, so that's another caveat that we might think we're sick while we're not anymore. Yeah, okay, this is, and then other tests there are several other tests also based on LAMP that, for example, Mammoth company has added another step, which is based on CRISPR, and that allows the detection on a lateral flow, but it also adds more time. So I think it's, as far as I saw, about 70 minutes, but maybe they improved it already. So, but I think there are many, many tests. Another one in Israel was based on the air. So although it's also based on antigens, the one I read about. So here the advantages are that it detects the existence of the viral RNA, nucleic acid. It can be applied to either other pandemics, other viruses, other pathogens, just needs to be adjusted according to it. So salmonella, other viruses, influenza, whichever, HIV. And it doesn't require a professional team. It can be applied at the entrance of the workplaces, at home, can serve as a surveillance. At this point, I wouldn't say it would be instead of the test, but it would allow to reach out to a larger population, and easily, cheap, etc. Yeah, so I would be happy to take any questions. As I mentioned, it's off track, but I hope we helped the world in our way, and we're still trying to see how we can turn this into a device so that it will be easy and cheap and applicable. Thank you so much.
Thanks, Naama. That was very illuminating, and certainly you have been helping the world this way. I'm going to turn the question and answers over to Tori. Are you there?
Yeah, I'm here. I have a couple of questions, but first of all, I just wanted to thank both of the speakers for 2 excellent talks. I feel like I learned a lot from both of them, and they were nice, short, and sweet, and to the point. We have a couple of questions on the question and answer format. One of the first ones that I saw pull up was actually from Peter Stambuk for Dr. Josef Penninger. So how much genetic variability is there in the human ACE2? And does this variability affect viral load at all?
There's quite some variability. I'm actually getting all these papers to read you now. Well, all the sequencing people and the big AI people also mining all data. And there's quite some variability. You know, if it's to the receptor binding domain of spike, if there's some variability in there, so it must affect it. So the question really is out, you know, if a lot of ACE2 is bad or if lots of ACE2 in the body is good because of this dual effect it has. But this variability, even here, there are people who are natural mutants for ACE2, which is quite interesting.
As a follow-up, do you think that any of that variability could be seen in, I guess, the variability that we're seeing within the COVID patients themselves and the symptoms that they're expressing?
Could be. There's actually also variability, very classic variability affecting ACE function. Of course, ACE drives Ang2. One of these changes in African Americans, which actually makes them not sensitive to ACE inhibitors. And what I understand in places like Detroit, so they're disproportionately severely affected. So one should not just look for ACE variability, but also variability in the whole pathway. Or ACE2 variability, but also for other variabilities in ACE and AT1 receptors and so on. And of course, then bradykinins come in because ACE2 also inactivates bradykinin. And that's and bradykinin B2 receptor is known to open up blood vessels for edema. That's actually one of the reasons why ACE inhibitors sometimes cause cough and actually edema in the lung because of this pathway. So yes, we should look at variability.
This is a comment more so than a question, but one of the attendees said, thank you to Dr. I'm sorry, Geva-Zatorsky.
Sorry.
It would be great to bring your method down to South America where the virus is expanding instead of Europe where it's already starting to slow down. You mentioned possibly making this like mass, almost like a mass testing method. Do you think there would be a way to kind of bring that into other areas as well? Is it difficult to produce this, I guess?
Yes. This is definitely high motivation. So we are actually having a couple optional devices shipped to us, 100 in number these days, and from from a company that, or yeah, a company, I guess, that manufactures them, and they might, I mean, we need to test them, but if it's possible to work on those, to marry, actually, when we talked, I actually use that word as well, to marry between these two technologies, then it's possible to have a very simple and under $5 device that can be spread massively to South America, Africa, places around the world, yeah, as well as to the Western world. As actually, it's also very important when we exit the lockdown phase.
Right, to keep everything. This question is for Dr. Penninger. You mentioned some sex differences within the infection, especially I think one of the areas that you saw a sex difference would be kidney damage, where males were exhibiting more kidney damage than females. Have there been any other known sex differences with treatment options? And
so sorry if I was unclear. So what I mentioned is in COVID-19, males are more affected in severe COVID-19, and actually also there's more mortality in males. So there's clear gender bias in the severe cases and the mortality cases. So the data shown was actually old mouse data, so mice, 12 months old mice, and the males got a spontaneous kidney injury, but females didn't. I just found this interesting from our old data that we saw this bias in old animals, you know, being male or female. So let's see how this explains gender bias. I'm sure, of course, there are many other issues of gender, and, you know, sex hormones affecting immunity, immune systems, and so on. But certainly an interesting notion that the ACE2 is on the X chromosome.
We have a couple of Facebook questions for Dr. Penninger as well. Are there toxicity or immunology issues with the solvable ACE2? And how are the ACE2 or virus complexes removed from the patient?
Yeah, very good questions. So from the 89 healthy volunteers and phase 2 lung disease patients, we know there's a very good safety profile. So it's quite good. Of course, COVID-19 patients is a total beast. So that's why we are just writing up this compassionate use case. It looks very promising. It does exactly what we expect it to do. One issue, of course, is indeed this question. So what happens is, you know, if our molecule soaks up the virus, would this, for instance, change the immune system so you don't get enough antibody against the virus? So our data actually show they still make the same amount of neutralizing antibodies in the same timeline as one sees in other patients. So where it really goes, we don't know yet exactly, but it clearly does not appear to affect the immune system to kick in to make neutralizing antibodies, which was really the things which kind of worried me. You know, if you soak it up, maybe then the immune system cannot engage it properly anymore. But it seems there's no worry about it. And we also see it, which is interesting, when we do, because we give it in an IV infusion. So the question was, of course, well, it of course protects body, but how does it get into the lung? So we did tracheal aspirates, and we actually see ACE2 getting into the lung. So probably because the blood vessel opens, so it can leak into there, which is also very, very important to know.
It looks like we have two other follow-up questions about this as well. How would soluble ACE2 affect individuals taking losartan for therapeutic purposes?
Yeah, there's a raging debate on, you know, ACE inhibitors and AT1 receptor blockers, and some people proposed we should all drop them, and then learned society said, you know, don't do this because ACE blockers, of course, good protecting from strokes and protecting the vasculature. I think we don't have really the prospective data needed to come to a conclusion how this works. There are actually papers published recently where they correlated retrospectively that ACE blockers actually beneficial, which kind of makes sense based on the pathway. It's very interesting. Again, it's retrospective, so we need some good prospective clinical testing. But it's certainly, there's no indication as far as I know now to really stop ACE blockers or AT1 receptor blockers.
And
already.
one, I guess, last question, because we're a little bit over time now. Is there a difference in ACE2 expression in age?
Yes, I hear the data coming out that, for instance, in the intestine, it changes over time, which is, of course, very interesting. And we also found in the intestine, when we knock out ACE2, it actually changes the microbiome, which is also quite interesting. I just want to mention it. And here the data already published, the COVID-19 is associated with changed microbiome. How this all fits together, I don't know yet, but I think there's some interesting connections.
Interesting, yeah.
Well, thank you. Excuse me. Thank you all, Josef, Naama, Tori, and it was a very, very enlightening and enjoyable webinar session. And I want to
thank you very much.
Yeah, I want to welcome the two panelists to take a look at the Society for Experimental Biology and Medicine, and hopefully we can get you to participate. Okay, thank you very much. And
thank you very much.
Hi, everybody. Nice to meet you all.
Thank you.